In this lesson, we'll be taking a look at the plasma or the cell membrane. So first of all, let's take a look at the functions. The main role of the plasma membrane is to separate the cell's interior from the external environment. So essentially you have two totally different environments within inside and outside of the cell. And in order to do this, it has to regulate what enters and exits the cell. So small molecules like gases can get through, some hydrophobic molecules can get through. If you remember that if these are phospholipids, the tails are hydrophobic, so it's a very large hydrophobic area, so it allows like molecules through. Very small polar molecules can wiggle through at times because the phospholipids aren't completely rigid, but large molecules and charged molecules cannot get through because of this large hydrophobic center. So this ability to let some things through and not is known as selective permeability. And this is based on whether they're large or small, charged or not, or their solubility, as well as what protein channels are present. So protein channels will let things through that would not normally be able to just go through the phospholipids. So the cell membrane is primarily made up of a phospholipid bilayer. And this is because there are phospholipids, as you can see them all here, arranged as a continuous double layer. And they're arranged such that all of their heads, which are hydrophilic, are on the outer sides of the layer, whereas all of their tails, which are hydrophobic, point towards each other. So you get this gigantic hydrophobic area on the inside, which becomes an impermeable passage for polar molecules and charged molecules. Notice they are not bound together, so they can move freely past one another, but they are squished nice and tight together, so it is tough for molecules to get through. So we often describe the cell membrane using the fluid mosaic model. Fluidity meaning that those phospholipids and proteins move laterally. As previously mentioned, they're not bound together, so they can slide past one another. And those components don't always stay in the same spot. They can move throughout as the cell membrane moves and bends. It's not a rigid structure. The mosaic part comes from the fact that it is composed of many different macromolecules. So even though phospholipids make up the majority of that membrane, there are proteins and other lipids embedded in there. There are carbohydrates as well. So it is mosaic-like in that it is not only one type of macromolecule. So what are those components? The primary component are the phospholipids, and their role is to act like a barrier. And because of their hydrophobic nature of the fatty acid tails, it causes the membrane to be impermeable to any water-soluble molecules and water in general as well. So if we take a look at how they're arranged here, we can also imagine them kind of spanning around as a cell, being almost like a sphere, with an aqueous solution on the inside as well as an aqueous solution on the outside, without water being able to get through very easily, and those water-soluble membranes either being kept on the inside or kept on the outside because of the phospholipid barrier. The reason it forms the barrier like this is those hydrophilic heads want to be in contact with the water on the inside and the outside, and the hydrophobic tails cluster together because none of them like the water. Next component of the membrane that we're going to take a look at is cholesterol. Now we've seen cholesterol before. It is the ring structure of a lipid, and its job is to regulate the fluidity of the membrane. So that allows the membrane to keep moving and make sure that it doesn't move too far apart or doesn't stay too rigid together. So when there are high temperatures, the role of cholesterol is to make the membrane less fluid, because as temperature increases, thinking about the way that molecules move, they would start moving more and more and more, meaning that there are going to be more gaps and more spaces, which kind of defeats the purpose of having that membrane. So what happens is the cholesterol molecules actually bend when it gets warmer, forcing those tails tighter together, allowing for less movement of those phospholipids. Whereas when it is colder or lower temperatures, they prevent the membrane from freezing. So they straighten out when it's cold, but they make sure that those tails can't get too close together to get too stuck so that they absolutely freeze up, and allows the membrane to maintain some fluidity. In plants, instead of using cholesterol, they use sterols, but they serve a similar function as cholesterol in animals. Membrane proteins within the membrane. There are two major types. The first type are known as integral proteins, meaning that they are permanently embedded in the membrane. Most of them cross completely across the membrane or are what are known as transmembrane. Some of them, however, do not. Some common examples would be insulin receptors and ion channels. Now, the reason proteins are so good at this is looking at the amino acid chain, polypeptide chain that makes up that protein. You can have a section that is nonpolar that will very easily and readily embed itself in that hydrophobic center of the membrane, whereas the hydrophilic amino acids could be on either end, keeping that protein lodged in the membrane. Peripheral proteins, on the other hand, are temporary proteins, meaning that they attach to integral proteins, or they penetrate partially into the membrane, but then they're just temporarily there and they will move on. So a peripheral protein can attach and can continue moving on. An example of this is the protein cytochrome C in the electron transport chain. It actually moves within the membrane from protein to protein to protein, but it is not permanently in one location. So there are many functions of proteins within the membranes. We can kind of break them down into 4 major classifications. The first being transport. As we've spoken earlier, many molecules cannot get across the membrane. Even though your cell needs them, they can't penetrate the membrane. So proteins put into the membrane can allow them through. Some of them will move through passively via diffusion. Some will require some energy, such as ATP. One example of a molecule that requires a membrane protein is glucose. It's far too large to fit through the phospholipids, so it needs a protein to get it inside the cell. The next major role is enzymatic activity. So proteins can be embedded in the membrane and actually act as an enzyme and catalyze reactions inside the cell. So the enzyme is always in that location of some membrane, substrates bind to the active site, and will cause a catalyzed reaction. Common example of this, insulin uses membrane proteins to catalyze their reactions. Signal triggering. So, for example, a signal molecule like a hormone could be found on the outside of the cell. That hormone doesn't actually need to get into the cell, but it binds with a membrane protein, which will then trigger either the release of another molecule. It could cause another reaction to happen. But what happens is that molecule itself doesn't need to get into the cell, but it causes a change inside the cell. And lastly, we can recognize them as attachment and recognition. So what can happen is these proteins can bind to cytoskeleton and can cause movement of the cell or portions of the cell, or they can self-identify a cell as a particular type of cell or belonging to a particular organism. So this is seen in HIV and as part of the cytoskeleton. So glycoproteins, as the word glyco implies, glyco means sugar. So it's a carbohydrate and a protein combined. A glycoprotein is a carbohydrate chain that is attached to a protein. So carbohydrate, protein, attachment together. They are found only on the outside surface of the cell or the extracellular surface. They are not on the inside. Their major roles are structural support, cell recognition, so recognizing cells as self or a particular type, and interactions between cells. So some examples would be mucins, or your mucus production. When they recognize things in your respiratory system that shouldn't be there, mucus can be produced, or your antibodies, recognizing things that are foreign and stimulating your immune system. Glycolipids, on the other hand, are still the carbohydrate chain, but this time they are attached to lipids, or more specifically, the phospholipids. They are also found only on the extracellular surface, and their job is cellular recognition and cellular interactions. Most common example of this is blood typing, the antigens that determine whether you have A, B, or O blood groups. So if you take a look at a blood, depending on what that carbohydrate chain is attached to the phospholipids, it can determine blood as different types. So take a moment now and make sure that you can label all of the components that were discussed in this video on your handout or on a diagram of your choosing.